Okay, so it's a time to start, and thank you very much for everyone to join. And I am Wei Yanpen, the Senior Director of Business Development at Shuimu BioSciences. So in today's agenda, I will give a brief introduction of our company in about 10 minutes. Then Dr. Xiaodani will present the technical advances and applications of cryo-EM. And just a little bit of housekeeping before we get started. So if you have any questions during the presentations, please type them in the Q&A box just in your control panel at the bottom. So we will have the time for the questions at the end. And please also note that our webinar is being recorded and will be public in next few days. Okay. So let's get started with who we are. Okay, I will show. Actually, our company was founded in 2017. We have established the first commercial platform of cryo-EM service in Asia. Our headquarters is located in the BioSquare Innovation Center at the Life Science Park in Beijing. We are devoted to transforming new technology from academia to industry to accelerate drug discovery. At the moment, we have around 40 employees. As a fast-growing startup, our team continues to expand. The majority of our employees has an advanced degree. Many of us are from top universities or institutes across the world. In addition, renowned scientists with strong scientific background constitute our scientific advisory board. Professor Hongwei Wang is a co-founder of Shuimu BioSciences. He led the School of Life Science in Tsinghua University from 2016 to this year. And before that, he was assistant professor in Yale University. Professor Yi Fanchen is currently an investigator of Howard Hughes Medical Institute and a professor at University of California, San Francisco. His lab focuses on three-dimensional structures of macromolecule by single particle cryo-EM. So, as the first commercial cryo-EM service platform in Asia, we are equipped with a state-of-the-art instrument. The newly Cryos G4 is in the process of installation. It is expected that we will start to operate G4 to provide service from the 1st this September. Now we are mainly using the Titan Cryos G3 for service. In terms of services we provide, we are aiming to provide a full service from sequence to structure from cryo-EM to facilitate the scientists all around the world. As you might know, cryo-EM experiment should start with qualified protein. So we provide custom protein expression as production service with fast turnaround time and competitive price. So when we have the sufficient qualified protein, we could start the cryo-EM processes, including negative stain, the grid preparation, data collection, data processing, the 3D reconstruction, and model building. Particularly, I would like to have your attention to our self-developed grid for better resolution. It is challenging to prepare robust and clean grids in high-resolution cryo-EM. Our self-developed graphene EM grid can partly solve the problems such as preferred orientation, strong background noise, and so on. In September, we will also have a webinar focusing on this special grid type. Please follow us in LinkedIn or sign up our newsletter to get informed about our latest webinar. So our cryo-EM service includes two packages. The first package is a starter package, which enables a faster screening of your samples. The starter package includes free consultation before the project, feasibility analysis, service assessment. We will deliver a project report containing optimal conditions for grid preparation and 2D classification results in starter package. If you would like to have a deep dive into cryo-EM to high resolution, Explore package will be a good choice. On top of starter package, Explore package provides you extra large-scale data collection and data processing till model building and model refinement. We will help you to achieve a refined model upon the results from structure determination. So, in conclusion, as a one-stop service provider for cryo-EM, we are aiming to provide fast, accessible, and affordable cryo-EM service so that the scientists all across the world could benefit from cryo-EM to accelerate the drug development. In one word, you provide the sequence, we deliver the structure. Shuimu BioSciences, your reliable cryo-EM partner. So we would like to forward your discovery by our extensive experience, strong scientific background, state-of-the-art instrument, and comprehensive service. So here are some of our clients. Although we were founded in 2017, we started to operate in 2020. Until now, we have collaborated with more than 50 pharmaceutical companies and research institutes. Please feel free to drop an email to hi@shuimu.com for any questions. And we are always happy to assist. Okay, so now I would like to introduce today's presenter, Dr. Xiaodan Ni. Xiaodan is a senior scientist in cryo-EM and structural biology at Shuimu BioSciences. She obtained her PhD degree at McMaster University in Canada in 2017. Afterwards, she went for postdoctoral training at MRC in the UK and NIH in US from 2017 to 2020, working on mitochondrial outer membrane, translocons, and Parkinson's disease. So her work had been published in more than 10 renowned journals. Now we turn the time to Dr. Xiaodan Ni. Hi, Xiaodan.
Hi, Wei-Yan. Okay, thanks for the introduction. So now I'll share my screen, right? Okay, I'm sorry for the delay. Hello, everyone. I'm Xiaodan from Shuimu Science. So today I will introduce the advances application of cryo-EM microscopy, and especially focus on examples of how resolution revolution can help in facilitate drug development, such as like small molecules or antibodies. If we look back to the history, we may find that scientific breakthroughs often build upon the successful visualization of objects invisible to the human eyes. And at the very beginning, people used the optical microscope, try to look at the morphology of bacteria or large organelles. And in principle, the highest resolution a microscope can reach depends on the wavelength of the microscope. However, because of the wavelength of the visible light and the magnification limit. So people has long been very difficult to look at the life's molecular machinery. So a microscope that can look at objects into a molecular or atomic level is highly divided. So for cryo-electron microscopy, I guess most of the people beyond the cryo-EM field start to know better about the cryo-EM. Starts from 2017, when the 3 scientists won the Nobel Prize in chemistry because of their contributions in the cryo-EM technologies. Cryo-EM technology emerged in the 1970s and 1980s and experienced several milestone technology breakthroughs until the rapid development in recent years. And the early cryo-EM milestone technologies is due to the contribution of these 3 scientists. Perhaps the most important step came from the work of Dr. Jacques Dubochet's group. He spent more than 20 years to try to figure out how to freeze rapidly a very thin layer of biological samples into a very thin layer of vitreous ice, because in the vitreous ice, sample can keep in its natural state without broken. And Dr. Joachim developed many of the computational procedures and image processing algorithms. And Dr. Richard Henderson developed low-dose electron imaging and obtained the first high-resolution structure of an integral membrane protein, bacterial low-dosing. So although we have these milestone technologies developed for, however, for cryo-EM, still doing some relative low-resolution structure determination work over 30 years. And the resolution cannot compete with X-ray crystallography. And it was even ridiculed for a time in biology. And in 2013, when the Dr. Yi Fanchen and Dr. David Julius from UC San Francisco solved the first high-resolution structure of triple ion channel, it really revolutionized the structural biology field, because it proved that we can use cryo-EM to solve a very important and small molecules, especially for membrane proteins that is hard for X-ray crystallography. And this achievement is due to the development of the electron detector, which is called K2 Camellar, because it's a high detecting quantum efficiency, DQE, and it improved the signal to noise ratio. And most importantly, the images taken by the movie mode, and each movie is made of stack of frames. It allows us to correct the beam-induced particle motion by aligning all these frames. And meanwhile, there's a lot of developments in the computational image processing programs, such as Reline, Free Align, EMN2, and CrossBart. And both the software and hardware development and revolutionized the resolution of the cryo-EM technology. So it made cryo-EM technology from the biology to a method really dominating the atomic resolution structure determination nowadays. So now it's coming to the resolution revolution, that is what we know. From 2013, the first triple ion channel was sold to 3.4 angstrom. And to 2015, the beta galactosidase was sold to 2.2 angstrom. And to 2016, the cryo-EM breaks 2 angstrom resolution value. The glutamine dehydrogenase was sold to 1.8 angstrom. And to 2020, the beta-3 GABA receptor was sold to 1.7 angstrom. And the mouse apophyretin was sold to 1.2 angstrom. So we really reached the atomic resolution. And with the development of technology, we also can solve some very small proteins, like smaller than 100 kD. In 2016, the acetate dehydrogenase was 93 kD was sold to 3.8 angstrom. And to 2019, the structure of the alcohol dehydrogenase 82 kD was sold to 2.7 angstrom. And the methemoglobin 64 kD was sold to 2.8 angstrom. And now the smallest protein, which is solved to a very high resolution, is stratabidin, only with 52 kD molecular weight, and was sold to 2.6 angstrom resolution. In addition to the resolution revolution, Perhaps the most important advantages for cryo-EM technology is we can capture the natural and dynamic state of the proteins, especially for those macromolecule compressed. For example, in the study of 50S ribosome assembly, when by capturing different conformation states during the assembly, we actually end up seeing this movie. For this movie, you can see clearly how the assembly of the central protuberance region happened. And a similar case in the human spisosome case, by capturing 3 different conformation states during the activation, we can see an ordered dynamic transition from the pre-catalytic B compressed to the mature C compressed. So now we have learned something about the advantages of cryo-EM technology, but how does the cryo-EM work? So as mentioned before, it starts from rapidly freezing. In the proteins in the solutions in all the different orientations, we put one of the dropped protein and apply in the grids and use the filter paper to get rid of the excess solution and fast freezing in the liquid ether. So we end up seeing a layer, very thin layer of vitreous ice, and in this vitreous ice, we can see different orientations of proteins. And this process can be done in the vitro board, well has a very standard control of temperature and humidity. And once the grid has ready, we can put the grids in the microscope and observe in the microscope. So in the microscope, when the electron beam transmit into the sample, the electron will colliding with the atoms of the sample and create some contrast. So here in the micrograph, we can see the particles, and each particle represents different orientations of the protein. And we took the same orientation of proteins and classified it into the 2D projections. And with different 2D projections represent the different orientations, we can reconstruct it into a 3D density map. So if the 3D density map, the resolution is high enough, we can see here, actually we can see clearly the side chain has resolved. So in summary, the single particle work through starts from the protein purification. So here, the proteins, including some soluble proteins, membrane proteins, and macromolecule compressed, or the single molecule and protein compressed, or even antigen antibody compressed. Then the protein sample can be applied the negative stand to check the quality of the sample and make sure the best quality of sample can be sent to the cryo-EM data collection and image processing to get a final structure. So in the following a few slides, I will briefly introduce these steps. So we first look at the sample preparation and negative stand. So when purify the sample, we need to measure the sample's composition, purity, stability. And if it's a compressed composition, it's better to know about the affinity between different components. And then we use the entry level of electron microscopies to check check negative stand for these samples in order to make sure the particle distribution, the particle stability, and particle size and shade of the protein. Then after negative stand, we evaluate the quality of the sample and send to the cryo-EM. As mentioned before, so we fast freezing the sample into a very thin layer of vitreous ice. And this layer of ice, we can see different orientations of proteins. And once the protein sample is ready, we can continue to do the image processing and imaging and data acquisition. So then when the data is ready, we will do the 3D reconstruction. Benefits from the development of different programs, we now have many choices for the image processing software. And for the image processing strategy, briefly, it starts from the image pre-processing and then 2D classification, 3D classification, and the final 3D refinement. And roughly, for the image pre-processing, we first, so the movable images collected were first applied for friend-to-friend and intra-friend alignment, and to correct the beam-induced particle motion. So this is the very typical images that we can see the particle trajectory after correction. So the micrograph after align and correction can be aligned and averaged. So from this micrograph, we can start to pick particles and classify different orientation of particles into the 2D classes. And after 2D classes, we will do the 3D reconstruction. So for 3D reconstruction, we always need to introduce a 3D reference. So in principle, a 3D object is considered to be consist of a set of 2D slices. So therefore, so a 3D reconstruction can be reconstructed through a set of 2D projections. So here, I won't explain too much of the image processing algorithms. Briefly, we use the projection matching and maximum likelihood approach to do the alignment. In which the maximum likelihood approach actually line and free align, we use the possibility weighting, and it deals well with the electron dose and drift. And for the final 3D auto refinement, the gold standard Fourier shape correlation was used and refined the two different models independently to prevent the overfitting. Then after the 3D classification, it's time to do model building. So for the model building, we first need to generate a static model. So for those structures already exist in PDB or similar structures exist in PDB, it's relatively straightforward that we can use the PDB to refine back the model. However, for those de novo structure determination, it is basically it requires a very high resolution and a match with a very good quality. So we need to generate use the second structure prediction to generate an initial model for the manually model building. However, benefits from all those AI-based programs. So now we can use some software like dtracer, like QuadNet, try to build a model automatically. And now we are now very familiar with the AlphaFold2 and Rosetta-Fold2. And this software even is more convenient for us to do the protein prediction. It is a good complement of the cryo-EM to revolutionize the structural biology field. So now we know the advantage and the workflow of single particle analysis. So the most important is what's the application of the single particle analysis and how the resolution revolution can help in facilitating the drug development. So in the following slides, I will use some examples to explain how this cryo-EM help in explain some mechanisms of the small molecule drug or the antibodies. So we all know that one crucial step for drug development is to find potential drug targets. And the revolutionized technology of cryo-EM make it possible to look at various of druggable targets, such as the GPCRs, membrane transporters, ion channels. These membrane proteins are very resistant to X-ray crystallography. And we also can look at some proteins like spike proteins, CT antigens as well. And once the high resolution target structure has identified, we can continue to look at the interactions between small molecules and target proteins to support the structure-based drug design. And meanwhile, the cryo-EM structure determination can use to identify the antigen antibody interactions, and it's good for epitope mapping to optimize the antibody design and to figure out the mechanisms of the differences of antibody efficacy. And in addition, the cryo-EM structure determination also can be used to characterize the virus particles and help the development of the vaccine. So first, we will look at some examples of how cryo-EM help in determining the mechanism of small molecule drugs. So we all are very familiar with the pandemics of the coronavirus 2019. So COVID-19, the virus, replication of virus needs to depend on the RDRP enzyme, the RNA-dependent RNA polymerase enzyme. So if we brought the RNA elongation or RNA binding, it will further inhibit the infection of the virus. So remdesivir is one nucleotide analog antiviral drugs, and was also used as the treatment of the COVID-19. So by the high resolution structure determination of the remdesivir and RDRP complex, we actually can clearly visualize the mechanisms of the remdesivir. So from here, for better display, I put the RNA in the active site of the IDRP, and we can see here the remdesivir assembled into the prime strand of the RNA by replacing the nucleotide of the RNA. And once it replacing the nucleotide of the RNA, it will immediately terminate the elongation of the RNA. And another very old drug, Shuimu, which has more than 100 years old, and it was used to treat the palliative diseases. And in biochemistry assays, they find that the remdesivir has more than 20 times potent than, the Shuimu has more than 20 times potent than the remdesivir. So in order to figure out these mechanisms, they also use the cryo-EM structure determination to figure out high resolution structure of Shuimu and IDRP. And we can see here is very clear, the two molecules of remdesivir, two molecules of Shuimu binding at the active site of the RNA and completely block the binding of RNA. So from this high resolution structure determination, we can clearly visualize the mechanism of these small molecules. Then we can have we can look at an example of GPCR protein. So we know that the GPCR protein comprises the largest family of the transmembrane receptors. And in clinical, like 30% of the clinical drugs are targeted to GPCR. And alpha-2 adrenaline receptor, alpha-2AR, a G protein coupled receptor, and in response to adrenaline and non-adrenaline. So, and another small molecule called dexmedetomidine is a highly selective alpha-2AR agonist and used as the anxiety reducing and sedative medicine. In order to figure out the highly selectivity of the dexmedetomidine, we need a high resolution structure of the small molecule bind to the alpha-2AR compressed. We know that the GPCR protein actually is very small, so it is challenging in cryo-EM. However, by with the binding of G protein and the stabilized of fragment of antibody, they were able to solve the structure into a 2.9 angstrom resolution. And at this level of resolution, we actually can see very clear the binding pocket of dexmedetomidine, and even the interactions with of the small molecules and the GPCR protein. So from here, we can see clearly that actually the dexmedetomidine has the similar binding pocket with the ligand adrenaline. So no matter the the function core of dexmedetomidine, the imidazoline ring of the dexmedetomidine and the methyl amino group of the adrenaline has attached well in this binding pocket. So it can stay well in this binding pocket. However, if we change the ligand to the isoprenoline, so this very large isopropylamine group will cause the repulsive interactions in the pocket, and it will block the binding of the small molecules. So this example tells us that the highly selectivity of the alpha-2AR of the small molecule is due to the structural feature of the small molecules. Then another case is, another GPCR case is PAC1 receptor. So the PAC1 receptor belongs to the secretin receptor family, and a novel activation is highly related to the migrant and making PAC1 receptor a potential therapeutic target. And there's two peptides targeted to PAC1 receptor. One is the neuropeptide PACAP, and the other one is the native peptide from the same fry, mesadilant. So these two peptide ligands have diverse sequence. However, they have the similar receptor activation function. So in order to figure out these mechanisms of these peptides in the ligand-induced receptor activation, so we need to have a high resolution structure of the peptide and the receptor compressed. So these two near atomic resolution cryo-EM structures, and we can see that actually these two peptides have different have completely different ligand binding modes. However, the extracellular domain of the PAC1 receptor can change its own conformation to accommodate the different binding mode of the ligand. And this structure provide important information for the structure-based mutagenesis and functional assay to identify the critical residues for the ligand-induced receptor activation. So these are examples of the small molecule drugs. And in summary, that if we have the structures of the target and the interactions of the small molecules, we can continue to do, and this information are very important for us to continue to do some molecular docking and the structure-based drug design. Then we can continue to see in the case of antibody development. So use the cryo-EM structure determination, we can identify the conformation epitope mapping. By the conformation epitope mapping, it provides lots of information such as the conformation of the antibody and the interaction between antigen and antibody, and the stoichiometry between antigen and antibody. And even we can do some structural validation of the modified antibody. So in the following slides, I will also have some examples to explain how the structure biology and structure determination help in identifying the mechanisms of difference of antibody efficacy. So this is a very typical case that we use the structural biology approach to determine the mechanisms of differences in antibody efficacy. So 4-1BB, also called CD137, it belongs to the tumor nucleus factor receptor family. And currently, there's 2 agonist antibody still in clinical trials. One is uradomide from BMS, one is utominomide from Pfizer. And these 2 antibodies actually have similar affinities to 4-1BB. However, they behave different antibody efficacy. So uradomide has certain effect in the rich tumors. However, it behaves obvious hypertoxicity, so the dose need to be controlled. And the utominomide has more favorable safety profile. However, it has weaker potency. So in order to figure out these differences, we need to have a structure determination of the antibody and the 4-1BB compressed. So from here, we can see that the 4-1BB itself actually consists of 4 cysteine-rich domains. And the 4-1BB ligand binds to the domain 2 and domain 3 of 4-1BB. And when we look at the compressed structure, we can see that the utominomide binds to the domain 3 and domain 4 of 4-1BB. So it actually is a ligand blocking antibody. And uradomide binding to the domain 1 of 4-1BB. And this binding position, this orient binding position kind of completely exposes its Fc fragment for the interaction of Fc gamma R receptor and enhancing the following ADCC effect. However, the binding position of the utominomide is kind of parallel to the cell membrane and close to the cell surface. We know that there's lots of factors around the cell surface. So it's kind of restricts the Fc domain interact with the Fc gamma R and restrict the following ADCC function. In addition, when we see this uradomide binding position, it will allow it cross-linking multiple 4-1BB and 4-1BB ligand and dramatically improve the potency. So we know that the 4-1BB ligand is the trimer. So this binding position is benefits for the later stimulation of the ADCC effect. So this is the case that we use the structural biology determination to figure out the differences in antibody efficacy. So now we can see another case. We use the cryo-EM structure determination to figure out and to guide the bispecific antibody design. So HER2 we know is a human epidermal growth factor receptor 2, and trastuzumab and pertuzumab are antibodies that targeted to HER2 and was used as the treatment of HER2 positive breast cancer. And in clinical, trastuzumab is in combination with pertuzumab for increased potency, to increase the potency. So there's many hypotheses about the synergistic activity of the combination use of trastuzumab and pertuzumab. One is with the binding of the two antibodies, it will enhance the affinity towards HER2. However, when they solve the cryo-EM structures, they find that the trastuzumab and pertuzumab actually combine to the HER2 simultaneously with no obvious contact. So it means that there's not the case that to improve the affinity by binding of the two antibodies. However, this high-resolution structure of cryo-EM give us some clues to the design of the bispecific antibodies. From the structure, we know that the two fat of the one antibody cannot bind simultaneously to the two epitopes. However, if we modify the hinge region and make this two fat more flexible, and we allow this two fat to bind the two apital regions simultaneously. And in addition, we also can collect these two fats with a rigid protein linker. It will also achieve the goal. And this method of design has been used in the design of the HIV antibody, and they prove that the potency can be increased to 2 to 3 times. And the last example we can see is use the structure determination to identify the stoichiometry and differences in antibody efficacy. So the antibodies targeting human antigen CD20 are divided into two groups, Type1 antibody and Type2 antibody, in which Type1 antibody can stimulate more potent CDC effect than Type2 antibody. So the rituximab belongs to the Type1 antibody, and it is a mouse-human chimeric antibody. And it was used for the treatment of non-Hodgkin's lymphomas and certain autoimmune diseases. And both obinutuzumab and ofatumumab belongs to all human antibody, and in which the obinutuzumab belongs to Type2 antibody and ofatumumab belongs to a Type1 antibody. They was used for the treatment of chronic lymphocytic leukemia. So in order to compare the different potencies of the Type1 and Type2 antibody, they use the cryo-EM structure determination method to solve the structures of the CD20 with all these antibodies. And it's very interesting that they find that all of the Type1 antibody actually form oligomers with the stoichiometry of 2 to 2, 3 to 3, or 4 to 4. However, all the Type2 antibody can only form like the 1 to 2 stoichiometry. So this oligomerization of the Type1 antibody will form the so-called seeding complex, and it will facilitate the IgG and CD20 concatenation. So when the concatenation, the Fc fragments can be, it will concentrate the Fc fragments and enhancing the following CDC effect. However, in the Type2 antibody, it will form a terminal complex. So it couldn't find this concatenation effect to further enhance the CDC effect. So from the structure determination, we know that the Type1 antibody has more potency than Type2 antibody, but there's still a question remained because both obinutuzumab and rituximab belongs to Type1. Because both the ofatumumab and rituximab belongs to the Type1 antibody. However, in a biochemistry assay, they find that ofatumumab has more potent than rituximab. So in order to explain these questions, they further improve the resolution and focus on only one CD20 and one Fab and one FAT. So they find that compared with the rituximab, the ofatumumab has the two Fabs has sharper angles between the two Fabs, and also these two Fabs has more freedom compared with the rituximab the two fat has some interaction between each other. So this freedom and sharper angle between the fabs makes it more, sorry for the distraction. So I will start from here. The last case of antibody development, I guess like some of the people didn't catch because there's a silence for a time and I didn't notice. So for the CD20 case, like we know that there's two antibodies targeted to CD20 and Taiwan antibody and Type 2 antibody, in which the Taiwan antibody can stimulate more CDC functions than the Type 2 antibody. And the antibodies like rituximab belongs to the Taiwan antibody. And it is a mouse human chimeric antibody and used as the treatment for non-Hodgkin's lymphomas and certain autoimmune diseases. And the obinutuzumab and ofatumumab belongs to the old human antibody, in which the obinutuzumab belongs to Type 2 antibody and ofatumumab belongs to Taiwan antibody. And it was used for the treatment of chronic lymphocytic leukemia. So in order to study the differences of the efficacy of these antibodies, they used the cryo-EM structure determination approach to solve the structure of the CD20 and these antibodies. And it's very interesting that they found that all the Taiwan antibody conformed oligomers with the stoichiometry of 222, 3.3, or 424. However, all the Type 2 antibody stoichiometry is only 1 to 2. The two antibody bind to one fat. So, and this oligomer of the antibody actually can form the so-called seeding complex. And this seeding complex is convenient for the IgG and CD20 to form a concatenation effect because they expose the Fc fragments and then concentrated the Fc fragments for the following interactions with the complements and effect and stimulates the CDC effect. However, all the Type 2 antibody can only form the terminal complex. So this structure differences explain why the different efficacy happens in these antibodies. However, there's still one question remains that both obinutuzumab and the rituximab belongs to Taiwan antibody. However, they have a different potency because the obinutuzumab has more potency than the rituximab. In order to figure out these questions, they focus on the structure and further improve the resolution of the structure. And they find that in the obinutuzumab, actually the two fab has sharper angles between these two fab. And this two fab has more freedom compared with the rituximab because in rituximab, the two fab has some interactions because it has the secondary epitopes here. And in this case of the obinutuzumab, more freedom fab as a sharper angle of fab can facilitate the forming of the IgG and CD20 conchyliation because the sharper fat will stimulate the Fc domain in more close with each other and enhancing the further reaction with the complement and the CDC effect. So through the structure determination, we note the stoichiometry of the antibody and antigen, and also we figure out the mechanism of the difference of antibody efficacy. So through all these examples, in summary, to answer many fundamental biological questions, including what we mentioned before, like using cryo-EM to solve the structures and answer the mechanisms of small molecule antibodies, we just need to look at the thing. And now we even can look at the thing at the atomic level. And all these achievements are due to the revolutionized cryo-EM technology technologies. So today, my talk is here. So, and thanks to everyone for joining the talk and welcome to the questions.
Thank you, Xiaodan. Thank you for your presentation. Yeah, So for the participants, we are sorry for the internet disconnections, but now it's all normal. So we have we have several questions. So the first one is from Shen. So ask, why do you use different softwares when data collection and classification?
Okay, well, actually, we actually, well, for one project for the structure determination, we actually try different softwares. And at the very beginning, well, we try Reliant, but sometimes when the other software like Crossbar coming, because the Crossbar is very popular for its very high efficiency of image processing. If you don't have large compute resource, and it is very benefits for this faster efficiency in Crossbar. So we can combine all these softwares just to reach the goal that you can solve the structure efficiently.
Okay, yeah, and another question, how do you fit the X-ray map into cryo-EM density map?
So I guess if you have the high resolution of cryo-EM density map, so first you were, just as I mentioned before in the model building, so first you need to have a starting model. So this, if your structure has the PDB in the PDB database, it will be quite straightforward. So you can use the PDB and doing the rigid body fitting into your density map, and then try to refine the model again, refine the model against your density map. So that is more straightforward. However, if you can't find any PDB structures in the database, it means that it is the de novo structure determination. So it requires you have a very high resolution density map, and also the quality of the map should be good. So then you need to have a like secondary structure prediction, and also by the help of now this new protein prediction software like AlphaFold2 or Rosetta-4. So you can have you can have an initial protein prediction first, and then you can use these models and refine your cryo-EM density map.
Okay, thank you, very detailed. And here is another question from Xiaodong. So for the structural parts of small molecules and proteins, how high resolution structures we need may guide CADD or virtual screening? Are there any constraints?
I guess we need to have a relative high resolution to guide the CADD and virtual screening. So so when we have the small molecules with the proteins, even the whole complex can reach a very high conformation, but sometimes we find that the small molecule binding pocket actually has very low resolution, and we can't see clearly the binding pocket. So it means that we need to improve the resolution of the whole complex as the goal. And then based on this high resolution structure, try to find whether the binding pocket have a very detail of the small molecule binding. Sometimes you can apply some other approach like the focus refinement, just focus on the small molecule binding pocket, try to improve the resolution there. And the most important, I guess, is before acquire data acquisition, that you need to make sure if you have a small molecule, your affinity of the small molecule and the protein is high. Because sometimes if you don't test the affinity of the small molecule, you try a lot of work in cryo and you still can't find the density of the small molecules. So to me, I guess the most important is the first step, you need to make sure your small molecule has the high affinities with the protein. And then you try to improve the resolution of the whole complex. And the last step, you try to focus on that small molecule binding domain and try to improve the resolution.
Okay, thank you. Thank you. And here we have a very interesting question that there is someone asking how to join our team. And yeah, please feel free to send your resumes to hi@shuimu.com, hi@shuimubio.com, and our HR team will contact you soon. Yeah.
You are very welcome if you are a master or even a foreigner, yeah, to join our team. Yeah, well, welcome.
Yeah. And we are recruiting now.
Yeah, even you don't have any structural biology background, but if you have a biochemistry background, we can train you to be a structural biology expert.
Yeah. So, yeah. So, is there any questions? If anyone has questions, you could type them into the Q&A box. And actually here, I have one question to Xiaodan. So Xiaodan, would you like to share your insight into AlphaFold 2 with us?
Well, okay. That is, as these 2 days, AlphaFold 2 is very popular. To me, actually, the most very close benefit to me is to do model building. So if you have a, well, if you, especially for those de novo structure determination, if you don't have any models, then nowadays, well, we manually build a model and it is very time consuming. So if I have the same experience that we use that more than 1 week or 2 weeks just to manually build a model. But if we have the AlphaFold prediction, we can just input the sequence and have an initial model automatically and use this model to refine your density map. It was very, very convenient. So this is the most convenient factor I can think of. Yeah, and also for, I know the AlphaFold and Rosetta Fold is very powerful, but we still need to testing what's the limit of these wells. And for those small proteins like smaller than 100 kD, I guess that is no problem. But what about those larger proteins that larger than 2,000 amino acid sequence proteins? So we still need to test the limit. And also for those compressed, I'm not sure whether the AlphaFold 2 and Rosetta Fold now can then predict the compressed the structure and also can including those very flexible structures. So I'm not sure the potential of the AlphaFold and Rosetta Fold, but I'm sure our testing, we will find the bug and also improve the function of the AlphaFold 2 or Rosetta Fold.
Thank you. Yeah, and so there is one question regarding how could you access the file. Actually, please just actually we will share these recordings later. Besides, if you would like to have the slides, please just drop an email to hi@shuimubio.com. I think the name is very easy to recognize and to memorize. It's hi@shuimubio.com. Yeah, just drop us an email. And if there is no questions, I think today's webinar will end here. And I hope this webinar will be helpful for all of you. And thank you very much for Xiaodan's presentation. Very informative and yeah, very impressive. Thank you. And also thank you everyone to join our webinars. Thank you, and please follow us in LinkedIn, in WeChat, or sign up for our newsletters. And yeah, we will keep you updated with our latest webinars, and there will be more sharing about the scientific content or the latest advancement of the technology. Thank you, everyone. Thank you.